Good morning, everybody. And first of all, I'd like to thank Dame Sally Davis for articulating so clearly the problems that we have with antimicrobial resistance, and also, I think, giving some optimistic insights as to the fact that things are possible, and I think we're beginning to get some insights into how to make the possible possible. And this morning's session is going to give quite a wide overview of different aspects of antimicrobial resistance and how we might be able to take an integrated approach to solve some of the problems. So it's a great pleasure to have as our first speaker Professor Mark Woolhouse from the University of Edinburgh. Mark is Professor of Infectious Diseases Epidemiology. He came to Edinburgh following stints in Imperial College, prior to that in Zimbabwe, Canada, and Oxford University. And he's primarily interested in population dynamics at the human and animal interface, which, as you'll discover, is very important in terms of how antimicrobial resistance can emerge and spread. So, Mark.
Mike, thank you very much, and it's a great pleasure to be here and to have opportunity to share some thoughts about One Health approaches to tackling AMR with you. A few years ago, some colleagues and I wrote a paper describing antibiotic resistance as the quintessential One Health issue. And one of the points we made in that paper is whatever action is taken to solve the AMR crisis that Dame Sally has just beautifully articulated will only be sustainable, hold on to the word sustainable please, if it's based on a sound understanding of the relative roles of people, animals and the environment in the emergence, spread and persistence of AMR genes. People, animals and the environment. That's what makes this a One Health problem, because all those compartments are intimately involved in the antimicrobial resistance ecosystem. So this talk, I'll elaborate a little bit more about the One Health approach, say something specifically about AMR in the environment with an emphasis on wastewater, and then switch briefly to talking about livestock and wildlife in slightly unusual setting, actually in urban Nairobi, but we'll come to that, and conclude with a few comments on the state of play. Now, there are lots of these sorts of diagrams that you see in front of you there in textbooks, in reports, in conference talks and so on, showing the interlinkages between those three compartments, the humans, the food animals, and the environment, and the way that antimicrobials are put into the system, of course, often overlapping or at least similar classes of antimicrobials in both humans and livestock, and how resistance and resistance bacteria and mobile genetic elements can move through that system via food, via manure, via sewage, via water supplies, or whatever. Now, if, like me, you did your early training in ecology, you look at a diagram like that, and you immediately know this is going to be a very difficult system to understand. It's very complicated, there are lots of feedback loops. It's not clear what you do when you start interfering with that system. For example, what would happen if you simply, as an experiment, stopped giving antimicrobials to food animals at all? Well, you'd expect that to have some beneficial effect on what we're really concerned about, which is clinical AMR in human patients. But it's got to filter through all those arrows to get there. It's not obvious what the magnitude of the effect might be. Or, to take another example, what if we stop humans being exposed to AMR in the environment? I mean, through water supply and poor hygiene, for example. Then, again, we'd expect this to have some effect on clinical AMR, but it's a complicated system. It's not easy to see what that effect might be. Dame Sally mentioned this report. I also think it's a very good report by the United Nations Environmental Programme, came out earlier this year, called Bracing for Superbugs. And they've been thinking about those sorts of issues too in the report. And there's a plethora of really complicated diagrams showing the flows of AMR in much more detail than the one I just showed you. And they also have some data. So this data shows estimated levels of water contamination and pollution with antimicrobials worldwide. And you can see immediately from that, this is truly a worldwide issue, but there are hotspots. There are hotspots in South Asia, there are hotspots in Africa. And the way the report talks about this is it says that there's good evidence that there is a relationship between this environmental pollution and the reservoir of resistance genes in the environment. The significance and its contribution to AMR globally, they mean clinical AMR, is still unclear. That's my emphasis. But they conclude there is still enough knowledge, we know enough, to implement measures to reduce the factors that influence AMR from an environmental perspective. So we don't know everything, but we know enough to act. Actually, I think that's a very good summary of where we are, and probably quite a good summary of my talk. One of the ways in my group that we explore these complicated systems, these ecosystems of AMR, is to write mathematical models. This is the simplest one we could possibly come up with, generated by one of my PhD students a couple of years ago, showing those three compartments and the way in which antimicrobial resistance might move between them. And there's a number of things you can do with a model like that. You can work out what's important. Which of all those parameters in that very, very simplified model most affect clinical AMR? And two of them are quite interesting. One of them is one I just mentioned, but the exposure of humans to environmental AMR, particularly through water and poor hygiene. That's an important one. The height of those bars on that graph says how important they are. And another one which you might not have thought about if you hadn't written the model, but how long antimicrobial resistance persists in the environment, whether it actually is an effective reservoir. If it is, it's going to act as a buffer, and that has important ramifications for the system as a whole. Hannah did another experiment too. She did the experiment I just mentioned in the previous slide. She said, well, what happens if you cut out antimicrobial usage in livestock completely in a system like that? What happens if you stop humans picking up resistance from the environment completely, just as a computer-based experiment? And the results are shown in that complicated graph there. All you have to notice is that on that plot, the red violin plots tend to be higher than the green ones. And what that says is that for many different scenarios Hannah explored, stopping AMR getting into the human population from the environment could actually be more important than stopping the use in livestock. Now, this is a very simple model of a very complex system. We're not in any way claiming this is right. It's simply just helping us think about the problem. And it's told us something to think about. This could be really important, perhaps more important than we previously recognized. So how do you go about looking for AMR in the environment? Well, one tool that we've been using in conjunction with Frank Airstrup, my colleague at the Danish Technical University, has been looking for AMR genes in wastewater. And we've actually, originally, we're developing this as a surveillance tool. And in blue here on this, in red here on this graph, we see the conventional way of doing antimicrobial resistance surveillance. Tends to target hospital or clinical patients, tends to be based on bacterial isolation and culture. Protocols, methodologies are somewhat variable, often very variable. Sampling frames are variable. So which resistances you actually look for. So whenever anyone's done any effort to try and collate this information, it's really quite hard. It's very, very heterogeneous. Looking for AMR genes in wastewater is different and in many ways much easier. This is the blue lines on this graph. First of all, we're sampling whole communities. The methodologies are completely standardizable. We simply extract the DNA, we sequence it, do metagenomic sequencing, and use bioinformatics pipelines to pull out the AMR genes. This is relatively straightforward, and it's very easily standardized. So we think it's got a lot of potential as a surveillance tool, and we need to know what's in there. So one way we've been using this is actually looking at what's coming out of our hospitals. And this is Megan Perry from my group, who incidentally was very much involved in the mold that changed the world that Sally spoke about in her talk. And what Megan was doing in this study was trying to take wastewater samples from one of the hospitals in Edinburgh. This turns out to be a very difficult thing to do for an old hospital because nobody knows where the sewage lines are. So Megan found herself spending a lot of time in looking at dusty files in janitors' cupboards, trying to work out where to sample. But she eventually located sampling sites from 8 different clinical units in the hospital. There are automatic water samplers that are used to extract water in a systematic manner. And what she ended up with, when she'd done all the analysis of these samples, was a profile of the resistomes coming out of these 8 clinical departments. And I just illustrate the sort of data there. So this is just divided up by genes for particular antimicrobial classes. And you can do a lot of analysis on this. One of the analyses that they showed, Megan and her colleagues showed, was that there was a relationship between the abundance of AMR genes in the samples and the usage of drugs in the departments. You'd expect that, but you're able to see it. And this is particularly strong for carbapenems and vancomycin, less strong for amoxicillin. You could also look to see if there's a relationship between the genes you're finding and the levels of clinical resistance reported in those departments from their own data. Good relationships for the staphylococci and enterococci, hospital-associated pathogens largely, less so for the Enterobacter. But you can see clear signatures of what's coming out of hospitals by clinical department. And we concluded from that, first of all, the obvious, that hospital wastewater is an important source of AMR in the environment, and that stewardship, particularly of the hospital-acquired infections and the carbapenems and vancomycins that you use in hospitals, would impact the burden of environmental AMR. So far, so good. So we then set up a different project that looked at this on a larger scale. This is back to Hannah Lepper's work. And Hannah had a wonderful sampling design. She chose 5 wastewater systems with hospitals in the wastewater system and 5 without, and compared what was going on coming out of the hospitals, what was coming out of the communities, and what was going into the wastewater treatment plants in that design. This was work done with our colleagues at Scottish Water, which is a new collaborator for us, but they were very pleasant to work with. A lot of this work, incidentally, was done during the height of the pandemic, and the few occasions we were allowed out, they were out sampling and managed to get the job done. So again, extract the DNA, do the metagenomic sequencing, end up with very large data sets describing what genes are present in what samples. And one of the tools we use to illustrate this sort of data are these 2D plots. It's just a data reduction method that allows you to get some indication of the makeup, the composition of the resistomes in these different samples. And what you can see very clearly from the left hand of those diagrams is that hospitals are different from what you see in the community and the treatment plots. Not so surprising, perhaps. What you also see is what you're finding going into the treatment plants is much more similar to what you see as the community as a whole. And that applies whether or not there's a hospital in the system. And on the right-hand plot, we allow the computer to do some machine learning on these data, and it could sort of just about guess whether there might be a hospital in the system at all, but it wasn't at all sure. Very difficult to detect a signal of the hospitals. So, conclusion is yes, hospital wastewater resistomes are different. They have more genes and more diverse genes, but the downstream impact isn't actually that great once you get to the wastewater treatment plant. Let's upscale again. This is some work that Frank Airstrup and his colleagues have been doing for a number of years, of turning this into a global surveillance system. And we call it the Global Sewage Project. And on the left-hand side, we see one of the outputs of this, and this is simply for 79 sites in 60 countries on a one-off survey, the overall numbers of AMR genes we're finding in the samples. So very, very crude, just as much as you could, simple as you could make it. And the surprising result for us there is in red. The highest numbers of AMR genes in the sewage samples are coming from Africa, even though we know that Africa has much lower usage than anywhere else. Europe is actually doing quite well on this particular measure. So Africa has higher than expected levels of AMR. On the right-hand side, we've expanded this to look at multiple samples from over 200 sites from over 100 countries. And what you can see there, I hope, is the beginnings of a global surveillance tool. We can start not only to detect, but also to monitor changes in abundance at these different sites over time. And there is enough consistency in that to suggest to us that we're getting actually a very useful signal from these sort of samples. This is an extraordinarily rich data set. I think it's fair to say that no one has ever been able to look at AMR in this way before. This is a new tool for trying to get a handle on the problem. And this is one of the things we can do to it. So another one of these 2D reduction plots, and now we're comparing not AMR, but the bacteria we're finding in the samples across continents. And the pattern I want you to look at there is the orange and the green areas, which are Europe and North America, are very similar indeed, and they're demonstrably different from everywhere else in the world. And that is something the microbiologists have known about for a long time. There is a distinct Western microbiome, and we're seeing the signature of that in the sewage coming out of these produced in these samples. So what about the resistome? Well, no one's ever done this for the resistome before. It's a very similar pattern. So again, Europe and North America are similar and mostly distinct from the other continents, with Africa in red looking quite distinct there. So what we have from that is evidence of a distinct Western resistome as well. Now, is that because different kinds of antimicrobials, different classes of antimicrobials are being used in the different continents? Well, no, because we can break all that down and do it one antimicrobial class at a time. And again, for most of that plot, if you start in the bottom left with the aminoglycosides, Europe and North America are very similar. And they're similar for macrolides, and different for everywhere else, similar for tetracyclines, a little bit different from the beta-lactams. But clearly there is something going on here that in different continents we are seeing different genes to the same antimicrobial classes. There is a continent-specific pattern to the kinds of resistance we're detecting. We can look to see what predicts those patterns. And we looked at a huge number of predictors, over 1,500 country-specific predictors from World Bank and development indices, using machine learning techniques, which mostly work quite well, although they had trouble with some of the data. And we get what you see on the right there, these heat maps showing where there are close correlations between AMR and particular predictors. I won't go through all 1,500, I'll give you the summary, which is this, that the strongest associations between the levels of AMR genes in these samples were with sanitation and healthcare variables. That was where the strong was, not with usage. Usage was not a particularly good predictor of what we were seeing in these environments. However, having said that, there was some heterogeneity from country to country, so if you did this in your individual country, you might find a different result. But what that tells to us is it's not just about usage, it's about transmission. In environments where there is poor sanitation, where there are weak healthcare systems, once resistance gets into the system, it's transmitting around, and that's an issue. What that makes you think of is WASH, water, sanitation, and hygiene. And for those in the audience who aren't familiar with these data, I think they're worth running through. Worldwide, over 2 billion people still lack access to safe drinking water. More than half the global population doesn't have access to safe sanitation. 3 billion people don't have access to hand washing facilities with soap, and hundreds of millions of people are still practicing open defecation. It's not surprising that we live in a world where once resistance gets into the bacterial populations, it's going to spread, and that's what it appears to be doing. So to go back to the sewage surveillance and the potential of this as a tool, I think it is a valuable tool, but there's a number of steps we need to do. We need to validate exactly how we collect these samples. We need to calibrate them against clinical isolates. We need to work out what populations we're actually sampling. We need to think about contamination. But I think there's potential here. I think we could set up quite straightforwardly some international standards for using sewage surveillance as an additional tool for a program like GLASS to monitor AMR. So I think we're far enough down the track that we can say that with some confidence. I hope it happens. Now, let's turn to food animals and their relationship with antimicrobial resistance in humans. Again, I'm sure we're all familiar that the intensification of livestock production is accelerating dramatically around the world as the demand for livestock products increases. If you look at that plot, some data that came out earlier this year, quite clear that the headline is right. Global use of antimicrobial centers on China, that big red dot on the map. And the next one actually is Brazil, which is increasing very, very fast. But what that tells you, if whatever we're doing to control the use of AMR in food animals, if China hasn't bought into it, then globally we're not going to have an enormous impact. That's where the action is in this area. Now, the O'Neill review a few years ago did a review of what they felt was the relationship between food animals and human AMR, and they did this by surveying the literature. Actually, they didn't quite survey the literature, they really surveyed the scientists, and they came out with a pretty clear consensus that there was a view that limiting use of antibiotics in agriculture would be a good thing to do. When you look at the actual raw data, that's perhaps a little less clear. There's an awful lot of papers on this now, and they produce a mixture of results. What I pick out from some of the meta-analyses is that you get the clearest associations between AMR in food animals and in humans when the humans you're talking about are not patients in hospitals, they're people working in the livestock industry. And that does seem to be a clear link. The one with patients is harder to pick out. For example, Sharon Peacock's group in Cambridge has done a really nice series of studies trying to trace a relationship between both bacteria and resistance in food animals to the patients they're seeing in their hospitals, and they haven't been able to find one so far. We did some modeling on this, a little bit like the modeling I showed you before a while ago. I'm not going to give you the mathematical results. I'm going to give you an analogy. Imagine your problem is not AMR in your clinic. Your problem is that you go into your bathroom, both taps are on in the bath's overflowing, and it's all over the floor. How do you solve that problem? You don't solve it by turning off just one of the taps. And that's actually a pretty good analogy for the way the mathematics works. Yes, just turning off the tap to antimicrobial usage in animals may not be as effective as you hope it would be, but actually, ironically, nor would just be tackling it in humans. You need to do it in both. This truly is an integrated One Health problem, and that's what I think the mathematics is telling us there. So you need to do both at the same time. So let's have a little closer look at this relationship between bacteria and AMR in people and livestock when the people and livestock are living in close proximity. And we did this in the city of Nairobi in a project led by my colleague Eric Fevre at the University of Liverpool and the International Livestock Research Institute. And Nairobi is a city, it's rapidly developing. There are a surprising number of livestock in this otherwise very urban environment, and in fact quite a number of wildlife as well. It's a very crowded place, everyone living in close proximity to each other. So we set up a very organized sampling design across the city. It covered a total of 99 different households, distributed carefully across different environments. We extracted over 1,300 complete genomes of E. coli we isolated from these samples, which is a very large sample for a whole genome sequencing project, about 10 times more than any comparable projects before. And you can make these nice phylogenies, and over the phylogenies you can overlay the properties of not only the bacteria, but also the sites they were collected for, including whether or not there's livestock, what resistance genes they carry, and all those sorts of things. And there's a number of analyses you can do on this extraordinarily rich data set. One of them is called core genome MLST analysis. I won't go into the details, but what you need to look at on that plot is a very clear spike in how many very, very similar bacteria you're finding within households. So there is a clear signature of a household here. The most similar E. coli are hugely overrepresented in the same household. And this is quite stable over a few years, so it's really quite an important unit for understanding the exchange of bacteria. At the next plot, we're looking to see exactly which E. coli are more similar to each other, and there you need to look at the red dots. And those red dots there are confirming, first of all, that you find the most similar E. coli in samples from the same household, but particularly from the same host type. So whether these are human-derived isolates, or livestock-derived, or wildlife-derived, that matters. So the more similar E. colis are overrepresented not only in the same household, but also in the same host type. So that's the bacteria. We can get some quite clear signals of how they're moving around, how they're shared in the environment of Nairobi. What about the resistances? Well, we can look at, again, using bioinformatics, the AMR gene abundance and composition in those three compartments, humans, livestock, and wildlife. What you need to pick out on that graph is that the largest bars are red. That means they're humans. So there's most resistance genes in samples taken from humans, less from livestock and less still in wildlife. And that's shown by the vertical black bars in this, so much higher levels in humans overall. The one on the left is by antimicrobial class. But you can also see on that there's a lot of variation. So we are seeing wildlife samples or livestock samples with very high antimicrobial resistance gene counts. But there is a clear pattern there. When you look at the composition of those genes, however, much less of a difference. So a huge amount of overlap between what we're seeing overall in humans, livestock, and wildlife. And we did another analysis too, which is these are all human resistomes, but compared with whether or not the household has livestock or not. And there's not a huge difference, as you can see. So that doesn't seem to be having an enormous effect. So humans have the highest levels of AMR genes, which is actually consistent for them to being the main source of antimicrobials in this system, although the livestock in particular are definitely treated, particularly poultry, with high levels of antimicrobials in that environment. But the resistomes across the different host types are really overlapping a lot. So there is a resistome of Nairobi that is quite clearly characterizable. We then went on, and this is work done by James Hassell at the University of Liverpool, to look at what we were finding in the wildlife, in the wild birds. That's a picture of marabou storks in a rubbish dump in Nairobi. James didn't get a lot of marabou. They're really quite hard to catch. But he did get a lot of samples from other kinds of birds, also mammals, but I'm talking about the birds here. And he developed this very simple model where the birds are really subject to spillover from human populations and indeed from livestock at the household level, and they are picking up their AMR genes that way. And what that plot shows, the thing you need to pick out, is the diversity of AMR genes in these wild birds is highest when you have a lot of people in the household and you have livestock. In those situations, you're finding a distinct signal that there's more diversity in the wild birds. He then went on to look at the data in a slightly different way. The vertical axis on those two plots is the level of multi-drug resistance in the E. coli from the bird. So these are wildlife carrying multi-drug resistant E. coli. And you can see what their relationship is with the levels of multi-drug resistance, actually the antibiogram length in humans or in livestock. You can look at it either way. The important thing about these graphs is that there are two different lines on each plot. For the relationship with human resistance, it matters a lot how the household manages its rubbish. You can see the picture on the left for the marabous. With the livestock, it matters a lot how the household manages its manure. So by this whole genome sequencing across E. coli across an entire city and looking at the household level, we can detect that spillover to wild birds is influenced by livestock keeping, by rubbish disposal, and manure management. This really is an environmental issue, and we can see the signature of that in these genome sequences. So where does this leave us thinking about the future of AMR? Well, I know other speakers this morning are going to speak on this first point, that there don't appear to be any new biotics on the horizon. But while we're waiting, the rest of us have to get on and think how we're going to manage this problem. Well, new technologies are clearly going to help, particularly diagnostic technologies and also preventatives. And the aim there is to reduce antibiotic usage and to focus it much more clearly. I think another particularly important area is education, educating our medics, our vets, this community, all of us, in ways that improve antimicrobial stewardship and again reduce usage. Those are two general points. But the one that I've tried to emphasize mostly in this talk is this one. We need to clean up our environment. We need to clean up our hospitals, we need to clean up our farms, we need to clean up our cities. And there are two benefits from that. Yes, you should reduce the need for antimicrobials, the demand from patients, but also, this crucial element, you lower the transmission rates. So you reduce the burden of AMR that's actually being passed through this whole system and ending up in clinical patients. Can we do that to a degree that's enough? Well, the point I want to make here is we don't get to decide. We as a community, the international agencies, national governments can set whatever targets they like about stewardship, usage, whatever, but it's the bacteria who get to decide whether or not that target is the right target. And this is where I do have one criticism of the UNEP report that otherwise I think is very excellent, but you'll find this in the foreword. Antimicrobials are an essential part of modern life. I think we'd all agree with that. Yet the more we use them inappropriately, my emphasis, the more the microbial world adapts. No, that's not right. The bacteria don't care what label we apply to the usage, whether it's misuse, excessive use, inappropriate use, or indeed essential use. It makes no difference at all to them. So the right phrase, in my view, is that word I started with right at the beginning of the talk, sustainability. Antimicrobials are an essential part of modern life, yet the more we use them unsustainably, the more the microbial world adapts. So my question for the AMR community and for this conference generally, generally that I'm going to leave you with is, what then does sustainable antimicrobial usage actually look like? How do we manage that very complicated AMR system to a point where the trends in resistance, the trend in drug-resistant infections, the trends in mortality that Sally alluded to at the beginning start to come down, start to flatten off and start to come down? I don't think we have a very clear picture yet of what sustainable antimicrobial use looks like, but I hope in these few minutes I've managed to give you some indication of the sort of tools we can use to gain that understanding of how this interacts across people, animals, and the environment to produce clinical resistance that ultimately we need to manage. And with that, I will thank a very large number of people. I've mentioned most of them as I've gone along, and an almost equally large number of different funders over many years of AMR research. And thank you very much for listening.
Mark, thank you very much. And we have time for some questions. And for those who weren't here before, if you have a question, can you make your way to one of the 4 microphone stations and let us know your name and ask your question. So, yes, please.
Hello, thank you for the talk. My name is Santiago Ramon from the University of Zaragoza in Spain. And I would like to ask you a question, actually a follow-up of your last reflection of the sustainability. We are all talking about developing new drugs, we need new drugs, novel chemical structures, but we have a large set of compounds that could be improved in the way they are being used. And I would like to know your impression about the repurposing approach. And I'm not talking about repurposing a cancer drug, for example, for antibacterial, but the same antibacterials actually used more wisely. And a follow-up conversation in the previous keynote talk about how to use combinations from these infections. We come from the TB field where combinations is the norm, but we see that in the treating gram-negative, gram-positive infections, this concept of using drugs in combinations that you can actually utilize more efficiently is not really fully explored. So I would like to know your impression about this repurposing approach in combination of drugs.
No, thank you. I think that's a very good point. And everything we can do to improve the usage of the drugs we still have and the stewardship of them is worthwhile. It always strikes me that still the most widely used drug in the world, antimicrobial in the world, is penicillin, despite the fact we've known about resistance for that for so many years. So I think that's a very important point that you raise, that we should be thinking about repurposing, reusing. And we have to be smart about this. Now, what I don't know, because nobody knows, is whether or not that will ultimately prove to be sustainable. But it's got to be more sustainable than what we're doing at the moment. So I think it's an approach to follow.
Thank you. Can we go to the back before Ashley?
Thanks, Joe Standing from UCL, and I've been on the antimicrobial stewardship team at Great Ormond Street for a number of years. One of the things that we've noticed trying to give feedback to prescribers is whilst we can tell them about use of antibiotics, we can only really feed back to them on phenotypic resistance, sensitive or resistant, which is obviously far too imprecise. So I'm very interested in this idea of measuring resistance genes in wastewater, and how can we get to potentially an automated way where this is sampled and sequenced, and we could give not real-time feedback but kind of regular feedback on the resistance genes that we're finding.
Technically, that would be pretty straightforward to do. Just a question of who does your metagenomics and who does your bioinformatics. These are all routine technologies. We actually did ours in Denmark because we happen to be working with a team in Denmark, but it would be perfectly straightforward for the UK to set up this sort of system. I think that is a feasible goal. I think the issues with it are not so much technical. I think those have been solved, but exactly what that information is telling you. I didn't mention the main weakness of the sewage surveillance, which of course is you cannot relate the genes directly to the bacteria. So the genes are there, the bacteria are there, but you can't match them up. We are working on that. The bioinformaticians are working on that. So there are ways you can go. So it's a very high-level overview, and it's different in nature from clinical assessment. But I agree with you. I think it would be a useful tool.
Thank you. Ashley.
Yeah, Mark, Ashley Stevens, Focus IP Group. Mark, I'm just going to make an observation that it seems to me the pandemic has emphasized the importance of wastewater sampling. I know in the Boston area, we get weekly reports in the Boston Globe of sewage samples from 3 places in the state. So maybe you can piggyback on that.
So actually, you are absolutely right. I think Frank and I were seen as slightly sort of mad advocates for this system in 2019, and SARS-CoV-2 has changed that completely. And now we're being asked by all sorts of people to come and explain the methodology to us. So there are very, very few silver linings to the pandemic, but one of them is now sewage surveillance, I think is firmly accepted as a public health tool. So thank you for mentioning it. Of course, we use it for polio as well, and other bugs, so yeah.
Can I just ask one question, Mark? It's, have we yet reached a stage where we know enough about antimicrobial resistance genes, their selection, the pressures upon them, to have a kind of equivalent of an R0 value where we might be able to see a reversal in the prevalence of those genes?
Modeling AMR, because R0 is always very intimately connected with mathematical models, is considerably harder than modeling a regular epidemic like COVID-19. And I think my cautious answer to you is I think the modeling community has some work to do before we could extract those types of indicators out of the modeling. It's a very active field, but we're not yet converged on the right way to approach that sort of problem. So put the modelers to work and get them to answer it for you.
Okay, good luck. Okay, any other questions? If not, I would just like to thank Mark again for a very lucid and clear introduction to One Health issues and antimicrobial resistance. And our next speaker is Professor Laura Piddock, who is here from the Global Antibiotic Research and Development Partnership, or GARDP. Prior to becoming head of research at GARDP, Laura had a long and distinguished career in studying antimicrobial resistance at the molecular level, particularly with respect to efflux systems. Laura has been a fantastic advocate for antimicrobial resistance, and she's been very important in various government initiatives, including co-authoring the O'Neill report on AMR. And Laura is going to talk to us today about global solutions to the antimicrobial crisis. And I think this rather follows from parts of Mark's talk, where we see that this is a global issue, where resistance in one part of the world will impact upon the problem elsewhere, and it has to be addressed from a global sense, and GARDP are leading the way in enabling that to happen. Laura.
Thank you very much. So, yes, I'm going to talk about the work that GARDP is doing, but first of all, I'm going to have some slides that really set the scene for what this organisation is doing and why. So, I think we all know before today, but particularly from the last 2 presentations, that antibiotics are the foundation of modern medicine. And I would challenge any of you to say that you've not had an antibiotic at some time in your life, and to ask yourselves, what would have happened if you've not had that treatment? There are some published works that indicate that antibiotics have helped extend our lifespan, particularly those of us that are lucky enough to live in high-income countries, by at least 23 years. So it's really hard to imagine a world without them. And that's part of the problem we are all challenged with, is that we expect antibiotics to be available. They are the panacea to all known germs, and that's something we have to challenge. So antibiotic resistance, as we've heard so eloquently this morning, is a global health crisis. It's not a silent pandemic, it's not a slow pandemic. It is with us, and it's been with us for some time. It's how we experience it that differs across the world. So superbugs know no borders, and you've just seen this already, and I'm just giving you one example here from some data from 2015, another global map, and the red here shows the countries in which this one resistance gene called NDM1 was distributed. Now, please don't think that those countries indicated in white means that this gene was not found. It's actually where they didn't look for it. And if we now look at data more recently, and I will show you some more maps in a minute, actually this resistance is globally disseminated. And I think the COVID-19 pandemic showed really well that microbes are invisible, they transfer very easily, we all move around the globe, our food moves around the globe, and we ourselves take our microbes with us. So NDM1 is just one gene that gives resistance to the carbapenem group of antibiotics. These are often called drugs of last resort. They're the ones that are used for those most serious life-threatening infections. And because of drug resistance, they're increasingly used around the globe. So it's a major global threat, and it frequently appears in all sorts of publications around the world. And of course, we ourselves experience it in the UK as well as abroad. So common infections are becoming more difficult to treat. And what do we mean by that? They're more difficult to treat? Because you and I, living in a high-income country, almost certainly, know that, very few people experience difficulties. But do we really know that? How often do we know friends and family have been told, ooh, that medicine isn't working, we'll try another. That one isn't working, we'll try another. And if it's an infection, that's almost certainly due to drug resistance. So it is here, and it doesn't matter where you are, but we are so fortunate in this country. We have a choice of different drugs available. They're accessible to us. And so most of the time, those infections are treatable. But as Dame Sally has just shown earlier, over 1 million people die of drug-resistant, antibiotic-resistant infections each year across the world. And it's not just the numbers that were shown in this recent report. They're considered to already be an underestimate, and there will be a follow-up report coming soon. We know they're an underestimate because if you actually look at the countries of which they sampled and got data from, it's not fully comprehensive across the globe. So if we take this as an underestimate, these are huge, huge numbers of resistant infections, either directly causing people to die or highly associated with people dying. And this is just one global map from the AMR review from a few years ago. And what I want to elaborate here is that the burden of resistance is not in high-income countries. It's in Asia and Africa. And that again was confirmed in that global burden of resistance study. So these are where the problems are. And as we know, what is somebody else's problem today is everybody's tomorrow. And if I can quote Peter Mugheni back in 2000 talking about HIV, where is the disease? The disease is where the drugs are not. And that quote is highly relevant to tackling antimicrobial resistant infections. So let's look on the left. I mentioned carbapenem resistant, and this is just 2 bacterial species here. And this shows, again, some maps. The hot colours are where the resistant strains have been shown to occur most frequently, so high rates of resistance. And it doesn't matter which of those 2 are shown, you can see that resistance occurs quite widely. But on the right-hand side shows where antibiotics are registered, in other words, where they're accessible to people. And unfortunately, the colours are the other way round. The hot colours show where they're least accessible. But you can see here that there is an association with where the drugs are least accessible with where there's most resistance. And this is something we need to really think about. So there seem to be misaligned priorities globally. The data says to focus on low and middle income country needs. R&D for high burden populations. I haven't showed this, but Sally did. You know, where is resistance most felt? Well, it's in children and newborn babies. That we really need to correct a public health failure, because we need to be looking at near-term development, access and appropriate use across all public health interventions. But this is what we're actually doing, and one could argue that's because it may be easier. We're focusing on high-income country needs. We're focusing on R&D for adults in those areas of the world. We're focusing our efforts on late-stage discovery to phase 1 clinical development, i.e. first-in-man studies, and a market failure. And now that's not to say these are wrong, but they're not encompassing all the global needs. So on the next few slides, I'd like to talk about what a non-profit with its partners can contribute to addressing the global crisis of antimicrobial resistance. And I divided this into 3 areas, and I am going to go through these reasonably quickly today, but these are all elaborated upon in our article that was in Clinical Infectious Diseases a couple of years ago. So the first issue is meeting that global public health need. And what are the interventions that are needed? Well, you need an objective and long-term approach, and you need targeted public-private partnerships so that you harness the best of the public and the private sectors. And you need to conduct R&D in different and sometimes challenging geographies where there's the highest burden of a particular infection, or in this case, the highest burden of antimicrobial resistance, to which you want to intervene. So the desired outcome is that new treatments address public health needs and are not just based on regulatory endpoints, so there's what needs to be done to get a drug approved for use, but or on commercial viability. And a non-profit can do this by focusing on populations and geographies from a public health perspective. The mission does require a long-term commitment for all involved, but can be less risk-averse to explore a potential product. The second issue is how to make the best use of public money so there's actually a public return on that public money. Indeed, we know that most products that reach the market for treating infections are supported by public money, irrespective of whether the organisation is a non-profit or for-profit entity. But with a non-profit, they can explore and invest in high-risk but high societal value projects and can expand access within low and middle-income countries. So they can aim to deliver the most needed treatments and bring novel interventions to those most affected in geographies that have little or no market potential. And this, of course, is why access has been to so few drugs over recent years. And with a non-profit, you can have this collaborative approach. And again, there's no commercial value. This is quite important. And local and multilateral approaches for R&D and access can be employed. We've seen, again, with the COVID-19 pandemic, we did not have equitable access across the globe to vaccines. And the same is true for antimicrobials. So how can you ensure equity and strengthen the global R&D infrastructure? And with a non-profit, you can help strengthen that network, again, in populations with that high burden or high need, and support the global networks and collaborations in these areas of the world, and really address underserved areas and deliver clinical trial data. And as just been mentioned earlier, that to get some drugs in some countries can take a very, very long time because you need to do clinical trials in the populations of that region. And one of the reasons for that is human beings don't always act the same in terms of healthcare facility, in terms of infections, what's causing the infections, in our basic physiology and biochemistry and distribution of drugs. We know that men and women are different in the way they metabolize drugs. We know that adults, children and babies are different. So all of these different populations have different development needs. And a non-profit can help strengthen research capacity in areas with high AMR burden. So non-profits can work on multiple projects with regional partners and importantly can be perceived as a neutral and trusted partner by both the public and private sectors and have the motivation and capacity to mobilise resources. So the Global Antibiotic Research and Development Partnership is a non-profit R&D organisation, and it was created in 2016 by the World Health Organization and Drugs for Neglected Disease Initiative. We were incubated by DNDi, and by 2018 we were sufficiently established that we became an independent Swiss foundation working globally. We are still in an alliance with DNDi and we work closely together on certain areas. So to give an indication of our global network, we have a GARDP North America, representation in Australia, we have a joint office in Southern Africa, and we have associated regional offices in Brazil, India, Japan, Kenya and Malaysia. And out of the Geneva office, people such as myself based in the UK work, but also Germany, Spain and further afield. So we are a truly global organisation. And we're also a broad church, so we have people from academia, such as myself, people from the healthcare sector, MSF, from industry. So we are able to bring great diversity and expertise to deliver our aims. So our vision is that all infections are treatable for everyone, everywhere. And to help achieve our vision, we work with partners to accelerate the development and access to treatments. So it's really worth reinforcing the point that we were set up to address a public health failure. We weren't set up to address a commercial failure. There are other activities ongoing to do that. And so the governance and partnerships that balance these perspectives are in a diverse set of countries and with diverse partners. And our strategy is based on disease burden and human cost. So we work to a disease area strategy. And again, reinforcing, we are working on addressing public health needs and access to novel drugs and providing the critical clinical data to support the use of new treatments and old treatments, which we've just alluded to a minute ago, where there's the greatest burden of disease. So we focus on three particular areas at the moment: sepsis in children and newborn babies, where 1 in 5 deaths are due to drug resistance. We know that over 140,000 newborn babies die each year of drug-resistant infections. That data is from the Graham study, which of course is an underestimate. We do also work on serious bacterial infections in adults, and one of the reasons for this is by partnering with companies that you need to get new drugs approved for use in adults first, and then you develop them for use in children and then babies. And we also work on sexually transmitted infections. So these are our 4 R&D programs, so not just the 3 I've mentioned to you. We also have a discover and exploratory research program. And the reason for this is part of our mission is to fill gaps that no other actor would fill. And we have this program to fill gaps that are not being covered, as far as we are aware, from publicly available information by national funding or international funding to academia or small companies. We have a separate external program to the R&D team called Accelerating Access, and this is to secure access to a portfolio of novel and generic antibiotics for everyone, everywhere. And then our 3rd area is called Scientific Affairs and Revive. And this is to capture and share scientific knowledge within the R&D community, because this community over the last 15 to 20 years has suffered a huge drain on expertise and knowledge and the wisdom of people who've been working in this field for 30, 40 years as they retire, or indeed as newer companies exit the field and that expertise is lost as they go to other therapeutic areas. We aim to capture that. So very briefly about each programme. So we're working with Venatorix on cefepime-taniborbactam, and that's finished a phase 3 clinical trial, which should go for a new drug application, and we hope that this will be approved because our role will then be to take this on forward for children and babies. And this, if approved, will be the first antibiotic treatment to be launched in collaboration with GARDP. We have an agreement with Shionogi and CHAI, Clinton Health Access Initiative, to facilitate access to cefiderocol, this is a new antibiotic, in 135 countries. And we have done an observational study. We've just started recruiting patients in 11 sites across India and South Africa to understand the management and outcomes of hospitalized patients with carbapenem-resistant infections, so that when we start to do some more clinical trials, we know where the highest burden of resistance is and can start our trial sites where that burden is highest. For children's antibiotics, this is what we're doing today, and we've mentioned combinations where we've already done the groundwork with teams to investigate what are the potential combinations that could be used to treat neonatal sepsis. We published recently in PLOS Medicine a study on over 3,000 babies from 11 countries, and we identified 3 antibiotics, they're old antibiotics, that we can use in combination. And this trial of using these combinations together will start soon. We're also supporting not only the pediatric studies for cefepime-taniborbactam, or we aspire to do that, but we'll also be doing the same with cefiderocol. So move on now to talk about our sexually transmitted infections programme. And I'm just going to halt because I have no idea how long I've got left to speak because it's still saying 30 minutes down there.
You've got about 12, 13 minutes to go.
Thank you. So, talk about zoliflodacin. This is an innovative oral treatment for gonorrhea. We've been doing this project in collaboration with Entasis, now Innoviva. And this is a drug that was originally from AstraZeneca, which was spun out into Entasis. So today, zoliflodacin has entered its final stage of clinical development, bringing us much closer to a new treatment for gonorrhea, particularly drug-resistant gonorrhea. So in March this year, we finished a major milestone for such a new organisation, having completed patient enrolment for this phase 3 clinical trial in 16 sites across 5 countries. Now, bearing in mind our organisation is only 5 years old this month, it's important to say that we were told you'll never get a phase 3 trial up and running. You'll never be able to do it. You don't know what you're doing. Well, we've done it. And that is a major success for us to actually done this trial and to have done it well. We really, really hope that it shows that this drug is efficacious, and we're waiting for the trial data to be unblinded and be available this autumn. So not only have we done the trial and working really in collaboration with Entasis and building on all the work that had been done before, but we've also successfully completed the remaining clinical trials to show bioequivalence and any drug-drug interaction studies and all the various non-clinical activities that are required to register this drug for approval. And we've developed and manufactured the final drug product for registration and commercializations in those countries in which GARDP would seek to make this drug accessible. And it's important to say that the access to this drug will be shared between Innoviva and GARDP in its different parts of the world that we're making it accessible in. So bringing together the clinical trial sites and the observational trials that we've been doing over the last few years, on this map here we can show all the sites where people have been working with us. So we've had the neo-observational study that I mentioned in babies, the carbapenem resistance study that we've just started in adults, and the solid fluorescein study sites. So we are well on the way to building our global networks. So I mentioned briefly discovery and exploratory research, and we've been very lucky to work with a variety of partners around the world, again shown on the map above, and we've just added 2 more organisations working with us, one in Brazil and one in Argentina. And what we have also been very fortunate is collaborations with several Japanese pharmaceutical companies who, whilst exiting infectious diseases drug discovery themselves, have continued to add new compounds to their libraries. And what that has done is they've shared those libraries with us, and we've been able to screen them. And out of this work, we've had 6 series of novel chemical entities, including 3 series of possible new antibiotics, and 3 more for potential to enhance the effect of old ones. And we just mentioned repurposing earlier this morning. One of those projects is a repurposing library to enhance the activity of a drug that's widely available across the world, including low and middle income countries. And this program of work has established a network of global partners that we're continuing to expand, and including working with Google DeepMind. So really exciting stuff that we're able to do with all sorts of different partners. So this diagram really shows where GARDP is acting at the moment with a few of the other actors in this field. This is not an exhaustive diagram. Just want to show here that there are a variety of actors working here and where we ourselves work. And just to point out that there is a lot of activity going on in specific areas, and what we each are doing is to complement each other, and we all need to work together to get new treatments to patients, because until we can actually solve the problems of sharing antibiotic-resistant microbes, we are always going to need new treatments. So finally then, capturing knowledge and sharing this with the R&D community. On the left-hand side, there's a wheel diagram showing you the very many different activities that we do. Particularly our Revive webinars, which are technical webinars that people participate in live with Q&A sessions. Everything we do is free and available on YouTube. We also have a variety of other types of webinars, AMR discussions and GARDP webinars. But I just draw your attention to resources. We have an antimicrobial encyclopedia. We use a lot of abbreviations and acronyms in this field, so if you don't know what something means, that encyclopedia should help you. That's supported by a library, and we will be launching later this year a roadmap, an interactive roadmap to tell you what goes on at each stage of the R&D life cycle. And we have networks and societies that work with us. And as you can see on the right-hand side, our Revive webinars really are picked up globally. We also, in collaboration with the British Society for Antimicrobial Chemotherapy, which is actually an international society, have an annual conference. This is virtual, deliberately, because that means it's free for everybody to join. It's live, but everything is recorded again. So if it's time zone challenge, people can still pick up the content. This year we did it with 2 organisations from Africa, last year 2 in India, the year before Germany, next year it's the Netherlands, and it will be early in February, so do mark your calendars, but everything is available at the website listed at the bottom. So the last thing I want to say on our resources is that we also work closely with an IUFAR project, Go to Pharmacology database, and we have a database called Antibiotic DB. It was something I started when I was still in academia, and I'm very lucky that GARDP decided they wanted to pick it up. And we have a poster on this at lunchtime, and I do hope you'll join us and have a look, and I can tell you a little bit more about these open access platforms to facilitate and advance antibiotic research. So thank you very much for listening to me, and on this slide I thank all of those that have funded GARDP so far. Thank you very much.
Thank you very much, Laura. So as before, if anybody has questions, please make your way to the microphones and ask from there. Perhaps I can kick off, and it's very interesting to see the antibiotic database that I'll definitely pop your poster if I can. And do you get some sense that within that sort of back catalogue of antibiotics which have been looked at but didn't make it, there may be some golden nuggets sitting there that will relieve some of the problems that we have now? We're perhaps less likely to be looking for sort of absolute total spectrum antibiotics and begin to pull these things out and look at them for individual or small numbers of bacterial infection.
Yeah, so one of the reasons I started the database was because very early, I would say probably 2003, there had been a discussion at Gordon Conference on antibiotic development where there was a desire that companies would open up their back catalogue of basically failed programs for two reasons. One, there might be some nuggets of gold in there, but the second one was to stop people rediscovering the wheel, particularly for things that were destined to fail for whatever reason. Unfortunately, that didn't come to fruition, so we started the database on the grounds that, well, let's see what we can get from publicly available information. So the idea then was, yes, to make it available for what can be picked up, particularly as, you know, chemistry synthesis has advanced. So some of the things in that database are from the 1960s or 70s, and we are continually adding that. What we found is that people wanted to know about newer antibiotics. So instead of it just being those that didn't succeed, before they got to IND, we now have them all the way through. We're continually adding that. And so, you know, it's really good that we link to the Go-to-Pharmacology database, because what that means is if you actually want to know more data, particularly pharmacological data, you can just link through to that where it's available. And actually Go-to-P links back to antibiotic DB too. So I really hope there's some nuggets of gold in there. We have had some positive feedback.
That's excellent. There's a question there.
Joe Standing again. Thanks very much for the nice talk. Just a quick clarification that NeoSep has started, and indeed the first PK arm has read out. We've got the data now, so it's really fantastic to see this focus on neonatal and pediatric infections. But my question is, NeoSep only tests 3 2x2 combinations, and of course, when we start thinking about combinations, there's many, many more that we might want to test. How can we fund these sort of, perhaps a platform trial that would, enable more of this to do? Because I think GARDP seems to be, from what I know, one of the only groups in this space that's actually trying to do this.
Yes, so clearly GARDP is focusing on babies in this context, but XACDURO is also looking at different clinical trial formats for adults, and their challenge is, where do you do the trials, how do you get the adaptive platform? And I think you are well aware that these adaptive platforms are themselves evolving, and the NeoSep trial that you just referred to has taken quite a lot of development and acceptance of the platform itself. And quite a lot of people would say, well, we'd like to look at more combinations. Can we plug this combination in? And certainly from some of other people's combinations, why are you not doing this combination? But as I'm sure you probably are aware, the combinations we're studying are based on the work we have already done, and we feel comfortable that those are the combinations we should be studying. But I think XACDURO are in a very good position to start looking at more adaptive type platforms.
Thank you. Any more questions from the floor? If not, then we are doing very well in keeping to time, probably a little bit better than our computer system appears to be doing. So I'd like to move on now to our next speaker, who is Dr. Darko Modun. Yes, please. Darko is from the University of Split in Croatia. He's a physician scientist. He's head of pharmacy at the University of Split and is interested in many aspects of pharmacy and pharmacology, including drug regulation and patient adherence. And Darko is going to speak to us about trends in funding of clinical trials for antimicrobials. And your abstract looked quite promising, so look forward to hearing.
Hello everyone, ladies and gentlemen. First, I would like to thank the organizers to give me the opportunity to be here. The title of my presentation is Number and Proportion of Phase 3 Drug Trials for Infections during the period of 2016 to 2021. I will keep my presentation short and sweet. A little bit about introduction, why, how, COVID-19 and what. Shortly about some methods, results and conclusion. So the question we are all struggling here today is why are there so few new antibiotics? As we know, resistance is growing every day on a global scale. For a few decades, R&D of new drugs of antimicrobials are in decline. And we can see that there are three types of obstacles: scientific, regulatory, and financial ones. Scientific obstacles, as we said before, there is an urgent need for new mechanisms of action for new antimicrobial drugs. Unfortunately, much too early in the drug life cycle, early development of resistance can occur. And finally, a drug can be too specific for their own good. So if you're too specific for a particular strain of bacteria, this is also very limited. From the regulatory obstacles point of view, only one but really important obstacle: what is the right endpoint of trial for antimicrobial drug? Is it total mortality, which requires high number of patients? Is it the clinical care rate, which is highly subjective, or is it something composite parameter? Finally, the elephant in the room: financial obstacles. Therapy of proper antibiotic drug, antimicrobial drug, lasts only for a few days, so we have a duration-limited therapy. Therapies, as I mentioned, is usually specific for a particular strain of bacteria, so you have market-limited therapy, and resistance inevitably occurs, so it's a race against time, so you have time-limited therapy. So the sad truth is there are better investment alternatives for a pure financial point of view, like oncology and diabetes mellitus therapy. So how to stimulate research and development? How to cure this problem? So you have push and pull mechanisms, you heard before. Push mechanisms usually require that you decrease the cost for the pharma company. Pharma company really like push mechanisms, like stack deduction, but general public really doesn't like that. The pharma company does not have to pay taxes when every citizen has to pay taxes. Pull mechanisms increase the price for the company when the drug actually reaches the market, as the Netflix approach in UK. So market entry reward. Of course, this is not so popular for pharma company. It requires much higher risk, so you have to put a new drug on the market. A few years ago, we published this paper about the factors related to COVID-19 pandemic that may influence antimicrobial resistance. I will not read all of this. So some factors that could increase antimicrobial resistance due to COVID. So increase to the halt of research, unlicensed use of some agents, self-medications, shortage of drugs and personal protective equipment, increased rate of periodical antimicrobial treatment, and of course, very popular one, overcrowding and overloading of the healthcare system. Factors that could decrease antimicrobial resistance during COVID: more focus on hand hygiene, social distancing, reduced travel, public's attention is now focused on infectious disease, and decreased antimicrobial consumption to fewer patients' consultation. And telemedicine actually might favor both increase and decrease in antimicrobial resistance. So what were our questions for this specific study? Did COVID-19 cause an increase in the overall number of phase 3 clinical trials for infections? And did COVID-19 cause greater inclusion of industry in funding phase 3 clinical trials? In other words, did COVID-19 cause a shift in number and funding of phase 3 clinical trials for drugs for infections? So it was a really easy, free, our favorite part, free study. Database was used clinicaltrials.gov to independent researchers. Results were exported as numbers and percentage of trials. So that was the time period, and countries we look were European Union, United States of America, and Japan. So the search points for clinical trial goal, interventional studies, status, phase 3, industry, and time period we looked. So European Union, United States of America, and Japan. So when we look at the European Union, we see that 2017 was annus horribilis, as the percentage of phase 3 from 17 and 18 declined to 2.7%. As you see, the number of phase 3 drug trials for all drugs increased to 752 to 2,668. On 2019, there is an increase, big jump from 2.7 to 10.9%, and slight decrease in the COVID years. When you watch now from the proper numbers of phase 3 drug trials for antimicrobials, you can see the much reduced number, like 34 or 49 in Europe. And you have this increase in 221, 24, and 228 drugs in the last 3 years. So just a big jump in the last 3 years. But you can see the percentage that are funded by the industry. So this have this increase in 2019, and then the decrease the 2 COVID years. So this is the story about European Union. Regarding the United States, again, you have a strong increase in all number of phase 3 drug trials, again, from 122 to 712. And the number of phase 3 drug trials for antimicrobials, you have different percentages. Again, you have a big drop in 2018, and then you have a nice jump. Also in 2020, you have 15% for antimicrobials. So again, when you see these numbers, you have 11 in 2016, and you have 83 in the last 2 years. So large increase of number of phase 3 drug trials for antimicrobials. And regarding the percentage, again, you have now increase from 57 from 2018 to now 72, 71 in the last 2 years. Regarding Japan, again, you have an increase in all number of phase 3 drug trials during these 6 years from 42 to 197. Again, you have big decrease, really low numbers of percentage of phase 3 drug trials for infections to 1.3, 1.9%. You have now a big jump increase in the last 2 years. Now you can see 9.7 and 10.2% of drugs are for antimicrobials. As you can see, the numbers show 1 study, 2 studies, 2018. Now we have 20 studies in 2021. And as you can see, in Japan, these studies are all more or less funded by the industry, so 100%, 90% in 2019, and back to 100% funded by the industry in 2021. So in the EU, proportion of phase 3 trials for antimicrobials varied from 2.7% in 2018 to 10.9% in 2019. Industry funded varied from 75% to 93.2%. In the United States of America, proportional trials for infections was its lowest, 5.5 in 2018, and greatest, 15% in 2020. In both European Union and United States, there's a trend of rising number of trials in recent years. This trend was also observed in Japan, where most infection trials are funded by the industry. Finally, to conclude, a jump in number of phase 3 trials can be observed in the past 3 years. However, a trend in increasing industry funding cannot be established in the studies countries. In all the country studies, there's an increase in the number of phase 3 clinical trials for antimicrobials, which is also, of course, really good news. And finally, in the United States and Japan, but not in the European Union, there's an increase in percentage of phase 3 clinical trials for antimicrobials, an increase in percentage of clinical trials for the treatment of infections funded by the industry from 2020. So some things have been changed by COVID, but not so much as we hoped for. Thank you very much. These are our team. This is like winter looks in Croatia in Split. This is me 5 years ago, before I invented the time machine. And you can see, have a chance to visit Split in a nice sunset from the medical faculty. So you are very welcome in Split in Croatia. Thank you very much.
Thank you, Darko. And there are any questions, please come to the microphones. And can I just clarify, the infections that you're talking about, is this all infectious diseases or primarily bacterial? Or
Infectious as they were registered in the clinical trial go.
And has the antiviral world dominated the last few years?
Yes.
Okay.
So,
And do you get any sense as to what's happening specifically for antibacterials over the same period?
so we didn't go into this subclassification at this point.
Okay. Any other questions? And just, I guess the other thing is that, so industry are also appearing to become interested, and perhaps there was a great incentivization that was driven by the pandemic. But do you sense from your studies that there are more companies appearing with a general interest in infection?
But there is the lag time. So in 2022, this is only about 2021. So I think that the last 2 years, you will see the proper results. So this is, of course, the limitation of this study. So I think that the trend is a good trend. Mm-hmm. So these figures will go up, hopefully, when we look on the last 2 years, 2022 and this 2023.
And do you think we'll sustain this trend at least for a few years if there's a global effort to try to prepare for the next pandemic, at least in antivirals?
I think that we can all hope for.
Okay, good. Okay, there's a question back there. Thank you.
Hello, I'm Chetna Desai from India. I was just wondering why we have focused on phase 3 trials. Would it have been, the picture would have been different if we also looked for phase 1 and 2 trials?
But we wanted to see what was the first step, last step before entering into the market. So, of course, you're right. Another good approach would be the phase 1, just to see what are the new ideas coming on the market. But we're just looking to see what was the last step before entering. But, of course, this is also a great idea for a new study.
Okay, thank you.
Okay. If there's no other questions, is that a question coming up here? Yes, one more, please.
Santiago Ramon, University of Zaragoza. I wonder what is the rationale underneath that most trials are funded by industry, and probably the question is because of the high cost of these trials, and that probably because all the industrial developers, they actually resource to CROs to actually run those trials. And going to the CROs actually increase a lot the cost of these trials. I'm wondering if there is any initiative, for example, at the European level, of public CROs that actually are funded by the governments and actually can lower the cost of this, all these trials.
So again, I mentioned the push and pull mechanisms. So some push mechanisms have occurred. But you can see the difference between countries. So you can see in Japan, it's more or less always 100%. So all clinical trials, phase 3 clinical trials in Japan, are indeed published by the industry. So there is no diversity. So it's really dependent on different markets, different countries.
Okay, well, Darko, thank you very much. And as we move ahead now to Matt Ronsheim from Innoviva, Speciality Therapeutics. Matt is, I guess, one of lamentably few CEOs of companies involved in antimicrobial development at the moment. He took over what was Epidemiology, and they have some pretty interesting looking compounds, one of which we've heard of just from Laura a few minutes ago. And Matt, who was a synthetic chemist by training, has spent a good few years now working in different pharmaceutical companies, specializing in antimicrobials. And he's going to tell us how innovation can avert the silent pandemic of AMR. But there's a caveat and a question at the end as whether we can sustain that. So, Matt, over to you.
Mike, thank you. It's a privilege to be here today. Thank you very much for including me in today's symposium. As Mike mentioned, I'm going to be talking about the innovation behind developing antimicrobials, but also, is it sustainable? Also going to have an opportunity to talk about the innovation behind developing the first pathogen-targeted therapy addressing infections caused by Acinetobacter. Just a little background on the companies. I've mentioned Entasis and Innoviva, just to give a little bit of history of the evolution of these companies. Innoviva is a healthcare royalty and asset management company, and they started investing in Entasis back in April of 2020. Entasis Therapeutics was a spin-out from AstraZeneca to really continue the antimicrobial and antibiotic R&D development in this space, and ultimately Innoviva purchased the remaining shares of Entasis Therapeutics last summer in July of 22. At the same time, Innoviva also purchased a commercial pharmaceutical company called La Jolla Pharmaceuticals. They have two marketed products, one Giapreza, the other is ribociclib known as Xerava. And ultimately, our three organizations have now been fully integrated to a full biopharmaceutical organization called Innoviva Speciality Therapeutics. This is a slide taken directly from the CDC. It lists the 6 to the 18 most alarming antibiotic resistance threats that are facing us today. You have vancomycin-resistant Enterococcus, carbapenem-resistant Acinetobacter, methicillin-resistant Staph aureus, carbapenem-resistant Enterobacterales, ESBL, and multi-drug-resistant Pseudomonas aeruginosa. We as an organization, really looking at how can we have an impact on AMR, we chose to try to tackle the problem facing carbapenem-resistant Acinetobacter. Why Acinetobacter? Acinetobacter is a nasty, nasty pathogen. Infections infect the lungs, bloodstream, urinary tract, skin, number of soft tissues. Infections caused by Acinetobacter have incredibly high mortality rates. Globally, is an average of a 30 to 70% mortality rate. Essentially, patients get infected with Acinetobacter, they have a 50% chance of dying. This is a nasty, nasty bug. It's considered a priority 1 critical pathogen as defined by the WHO, and is the 5th leading cause of death associated with antimicrobial resistance. Carbapenem-resistant Acinetobacter is also listed in the CDC in the top 5 urgent public health threats. And there's no clear standard of care in order to treat infections caused by carbapenem-resistant Acinetobacter. How do you approach tackling a bug like this? Typically, antibiotics are developed broad spectrum, broad label. You're looking at a number of different pathogens that the antibiotic may be able to treat and may be able to combat. Can we change the approach in developing a pathogen-targeted antibiotic, where you're delivering the right drug at the right time with the right dose? It's a very innovative approach to antimicrobial development. You're looking at PK/PD modeling in a very different way, where you're targeting a specific bug and you're dialing in what's the exposure I need to kill that bug. Can you develop an innovative clinical study with a really streamlined regulatory pathway? How long would a clinical study like this take? Nobody knew. My predecessor, Manos Peros, is a person I have tremendous respect for, was constantly told, you'll never be able to run this clinical study. How are you going to chase a pathogen all over the world and be able to accomplish this in a reasonable amount of time? And then finally, how much is this investment going to be? If you can't figure out, really understand how difficult or challenging it is going to be to conduct a clinical study like this, what's that going to cost? So developing a pathogen-targeted antibiotic required innovation across the entire development portfolio, or the entire development pathway. Rational drug design. You need an appropriate beta-lactam, beta-lactamase inhibitor combination that could be specific for Acinetobacter. The beta-lactamase inhibitor needed to be unique and differentiated in a way that was broad spectrum. PK/PD and dose justification. How do you dial in the specific dose needed to target Acinetobacter? How do you get to the exposure levels that we need? It's going to be specific for this bug. Manufacturing. It's a streamlined development program. Incredibly complex chemistry. You need API needed for preclinical development work, tox studies, in all of clinical development. You need a rapid optimization in the early stages, and also rapid optimization of cost of goods early on in order to satisfy a sustainable development program in a streamlined setting. And then a streamlined clinical and regulatory approach. It's a global clinical design. It's a very different phase 3 study design versus the typical broad spectrum antibiotic. It's a single phase 3 pivotal study. Because of the smaller patient populations, a larger non-inferiority margin is necessary in order to complete. With that said, I'm going to talk a little bit about the story of XACDURO. This is the first pathogen-targeted antibacterial approved by the US FDA to treat hospital-acquired bacterial pneumonia and ventilator pneumonia, bacterial pneumonia caused by susceptible isolates of Acinetobacter baumannii calco-acidicus complex. We just received approval in May. I'm going to tell you a little bit about the story of the discovery, the development, and how we got to where we are today, and hopefully where we're going in the future. So starting with discovery, you needed an appropriate beta-lactam, beta-lactamase inhibitor combination. SUL-Bactam has been around for decades, typically marketed or dosed as a beta-lactamase inhibitor, oftentimes in combination with ampicillin. What's unique about SUL-Bactam, while typically it's used as a beta-lactamase inhibitor, as a beta-lactam antibiotic, it actually has significant potency against Acinetobacter. So this is really important, identifying beta-lactam or an antibiotic that actually has activity against Acinetobacter. Problem is Acinetobacter is a smart bug, just like most other bacteria. It develops resistance to SUL-Bactam readily, and resistance mechanisms are quite predominant. So we needed to identify a beta-lactamase inhibitor that was broad spectrum and had the ability to fight the beta-lactamases that are produced by Acinetobacter. So the team had identified Durlobactam, which is a member of the diazobicyclooctane class of compounds, or DBOs for short, which is a common beta-lactamase inhibitor class, Avibactam, Relebactam, and others. The unique and differentiating properties of Durlobactam allow it to be broad spectrum, unlike the others. So what was important, Durlobactam has the ability to inhibit class D beta-lactamases, which is what predominates in resistance mechanisms for Acinetobacter. So Durlobactam effectively restores the activity of SUL-Bactam and able to fight and knock down bacteria, Acinetobacter pathogens. So continuing on in discovery, just some preclinical work to really show the potential efficacy of a SUL-Bactam-Durlobactam combination. You can see here we were evaluating the combination of SUL-Bactam, Durlobactam against extensively drug-resistant strains of Acinetobacter infections in both a lung and a thigh model. So talk first about the thigh model. You can see with the combinations of SUL-Bactam, Durlobactam, and in the green is measured against colistin, which was also evaluated in the study as a control. You can see the dose proportional effect of the combination of SUL-Bactam, Durlobactam in this study, and the effectiveness at which it knocked down the bacterial load of Acinetobacter in vivo and in our preclinical studies. You can see colistin as a control also did a decent job of knocking down the bacterial load. At the same time, this colistin was dosed at the maximum tolerated level. While it did do a good job, it didn't do as good of a job as SUL-Bactam, Durlobactam in these preclinical studies. Moving over to the lung model, you can see SUL-Bactam, Durlobactam still had a dose proportional reduction in bacterial load. In the case where we also evaluated colistin in this model, you can see colistin could not make it to the lung, so you could not get lung penetration, which was very important. But it was really important to be able to see that SUL-Bactam, Durlobactam could get the penetration we needed, or potentially, in the preclinical in vivo models. So this is a great start. Not getting into a lot of the other data, obviously, that was generated preclinically, but it was a lot of these results that we were able to use in order to try to predict or work to predict what would be our human efficacious dose for SUL-Bactam, Durlobactam. Moving on to development. Been in this industry for well over 20 years. I won't say how many more than 20 years, but well over 20 years. I've had the privilege to contribute to the discovery, development, and commercialization of now 8 commercial products. I have to say, XACDURO is the most challenging program I think I've worked on in my career, but also the most rewarding. The chemistry of durlobactam is incredibly complex. I won't get into a lot of details of it, but some of the importance with the innovation early on in the program is making sure we can get material. So chemistry for durlobactam was working on, it was an 18-step linear synthesis in order to produce API in a 1% overall yield. So chemists would be working, they'd be starting with kilos of starting material, at the end of the day end up with 100 milligrams of material by the time they got to the final API needed for preclinical work. Herculean efforts in order to get API in the early stages of this program. There's a lot of work that went into getting to something that could just be clinically viable, being able to get enough material, the innovation to get enough material to support a clinical program. So there were a number of improvements made. Synthesis was reduced to 11 steps from 18, and improvements to 18 to 24% overall, and a reduction in lead time of approximately 6 months for API. During the clinical development, there was obviously an extensive amount of work in order to make the manufacturing NDA ready and commercially viable. We won't get into the details of that here, but happy to talk about some of that later on, some of the breaks. But ultimately to get to a commercial package, and this is XACDURO, which is our commercially marketed packaging for this product. Continuing on development, a streamlined clinical development program. So we did follow a recent FDA guidance, treatment of serious bacterial diseases. Guidance for industry came out in August of 2017. I'm going to quote the FDA. I would not be able to say this, but since the FDA was able to say this, they wrote this in our briefing book for the advisory committee meeting we just had in April. The SUL-DUR development program is the first example of a streamlined program for a targeted therapy for a high unmet need pathogen, namely CRAB-C. This is a quote directly from the FDA. The strategies associated with clinical development and the regulatory development with regard to developing a pathogen-specific antibiotic are unique. You're dealing with a much smaller patient population than you would deal with in a broad spectrum typical phase 3 program. It's a single phase 3 study, not multiple phase 3 studies. We're dealing with patients, not only are they affected with Acinetobacter, which is a horrible infection, there are also a number of other comorbidities associated with these patients. You're dealing with very, very sick patients. The primary endpoint for our phase 3 study was 28-day all-cause mortality. Basically, after 28 days after infection, did the patient survive or did they not? It's an incredibly complicated clinical program. Regulatory strategies that obviously coupled with the clinical program, equally complex. You're dealing with, again, it's precedent-setting clinical study design. We continuously discussed our positions with the FDA. The FDA discussed their positions with us, and we wanted to maintain alignment with the regulatory agencies all throughout our clinical program. Our phase 3 study, again, it was a single phase 3 study, global program. You can imagine the complexity of trying to identify clinical sites all over the world that potentially had the highest probability of Acinetobacter infections in their hospitals. So we had 71 sites. They had the ability to screen patients across 16 countries. 59 of those sites were successful in randomizing patients into our clinical study. And a couple things I want to mention is, as I've said, is an incredibly complex clinical study design. I want to add that the clinical sites were in hospitals during the COVID-19 pandemic. You could imagine the complexity associated with conducting a study such as this one in the middle of the pandemic. I'll also add, we were able to continue supply, manufacturing throughout the study uninterrupted. And everyone's familiar with the supply chain issues that took place during this period. We're incredibly proud of the fact that we were able to complete this study in less than 2 years. Now on to the results. Like I mentioned, our primary endpoint was 28-day all-cause mortality. You can see on the left, XACDURO had an all-cause mortality of 19% compared to colistin, which was 32%. One thing I forgot to mention, I'll add, in developing a pathogen-targeted therapy for Acinetobacter where there's no standard of care, how do you choose your comparator? So colistin, I think most are familiar with, is a nasty, nasty drug, but it's also the most prescribed drug used to treat carbapenem-resistant Acinetobacter. And you can see here the efficacy results, and it's worthy to note, like we had mentioned, because it's a small patient population, it's pathogen-targeted and streamlined, our non-inferiority margin was set in agreement with FDA at 20%. A typical phase 3 program, your non-inferiority margin is set at 10%. I'm sorry, 20% what we had is normally set at 10%. Even though we had the small patient population size, we still met the 10% criteria. And we'll move on to the next slide to show us more of the consistency of the results. So this is moving on to the secondary endpoints. Up in this forest plot, the top primary endpoint category, you can see 28-day all-cause mortality. You can see towards the left favors XACDURO, whereas towards the right would favor colistin. What's remarkable about our data set is the consistency across the board. You can see in all the secondary endpoints following 28-day all-cause mortality, 14-day all-cause mortality, in crab-infected patients and non-crab-infected patients. The consistency of the data is remarkable. The consistency of that data also carries on to microbiological outcomes as well. The clinical micro was just as consistent as our efficacy data. It's important to note that the dotted line you see at the 0 point, that would be a measure of superiority. And for those that work in, I see a head nodding, in this space, know developing something in the antibiotic space as superior is incredibly rare. You can see how close we were. Quite remarkable. Yes, that's right. So we're incredibly proud of this data set and how we were able to accomplish it, and the innovation that went into it. Moving on to clinical cure rate. And we define clinical cure rate as complete resolution or significant improvement of baseline signs and symptoms and no new symptoms such that a new additional gram-negative antibiotic is warranted. You can see here the clinical cure rate with XACDURO was 62% compared to colistin, which was 40%. Moving on to safety. XACDURO, or SUL-DUR at the time, sulbactam, durlobactam, was well tolerated and a favorable safety profile across the entire clinical program. Here we're just talking about the phase 3, but the entire clinical program. The other primary endpoint that was achieved was in safety. SUL-DUR achieved the primary safety objective of lower nephrotoxicity than colistin. I think everyone's familiar with the nephrotox associated with colistin. In this case, we were actually superior statistically. The most common adverse events with this product are typical liver test abnormalities, diarrhea, anemia. And it's also safe to say and consistent to say that XACDURO, safety profile for XACDURO was consistent with other drugs in the beta-lactam, beta-lactamase inhibitor class. So we've talked quite a bit about the innovation in discovery, in development, and ultimately the results of a product that can cure a number of patients with this infection and save a lot of lives. What was the investment to get there? So this is the timeline of what our high-level clinical program looked like. Entasis was spun out from AstraZeneca in 2015. And you can see here, we went through the IND-enabling studies and preclinical development. We ran 6 phase 1 studies, in addition to a phase 2 study between the years of 2017 and 2019. And then starting in 2019, we initiated the phase 3 pivotal trial, the ATTACK trial for XACDURO. You can see in the costs here, we totaled approximately $250 million investment in order to bring this drug through preclinical and ultimate clinical development into a fileable package. Question is there going to be a return on investment? We've heard a lot of people talk today about the investment required and being able to be in a position where a return on investment is viable. Large pharma has primarily moved away from this space, moved away from the innovation because of the lack of return of investment. Number of our peers, it's been spoken earlier, have gone bankrupt, developed interesting products, innovative products, but have gone bankrupt because of the lack of return on investment. There are a number of ways to approach this. Government legislation. There are a number of acts that are in legislation now, and unfortunately have been in legislation for quite a long time. We have to get them to the point where they cross the finish line to help. So we have a big problem that we're going to be dealing with. And the innovation is there, but we have to make sure that companies are incentivized to participate and continue to innovate and drive. Another area to consider is the different ways to commercialize. So typically antibiotics have been profitable in the past by being high volume, low cost, broad spectrum products. We've taken a very innovative and a different way where it's a pathogen-targeted approach where you're developing a product that you can deliver the right drug at the right time, at the right dose in order to cure an infection. This precision-targeted approach is innovative and novel in the antibacterial space, but it's not necessarily innovative and novel amongst other areas in pharmaceuticals, oncology, rare disease, orphan disease. So I'm going to put a few numbers here. I'm going to preface this. These numbers are for US. Right now, there's an estimated 15,000 to 25,000 cases of crab in the US per year. There's two things to that. Globally, you could probably multiply that number by 30 at least. But in the case of the US, I think these numbers are severely underreported. Regardless, when you're dealing with small numbers like this, the classification for orphan drug designation is less than 200,000 cases per year in the United States. So clearly this fits well within that. The mortality rate of patients that are infected with Acinetobacter is 30 to 70%. And it's important to note that this is not mortality over months and years. This is mortality over days and weeks. There's no clear standard of care. So how do we establish in the antibiotic space a pricing structure that makes this business viable? It's a viable business in other disease areas. How do we make it a viable business also in driving innovation and really incentivizing investment in innovation in the antibacterial space as well? So there's the government policies, and there's also commercialization paradigms that also need to evolve as we develop these innovative medicines in order to address antimicrobial resistance. So in summary, we have developed an innovative approach to addressing an urgent public health threat, specifically hospital-acquired and ventilator-associated bacterial pneumonia caused by Acinetobacter baumannii calcoaceticus complex. It's the first example of a streamlined development program for pathogen-targeted antibacterial, namely CRAB-C. While our work was incredibly successful, it required a huge investment, a very large investment. It was extremely challenging in order to get that investment financing in order to continue and complete this program. We do need a new commercialization approach in order to continue this innovation and make it sustainable across the board. This approach, however, does establish a precedent for future precision-targeted antibiotics to address the AMR crisis that we are facing today. Acknowledgements. One, I'd like to thank the entire Innoviva Specialty Therapeutics team, including past and present members of Epidemiology Therapeutics. I wanted to share this picture. It's a very proud moment for us. This was the moment at our advisory committee meeting. This is our team. And you can see on the screen behind, this was when the FDA flashed up the unanimous vote for recommendation for approval of 12-0. And you can see how excited our team is. It was a fantastic moment for all of us. First of all, preparing for that meeting, which was incredibly rigorous preparation, but just the success of that meeting and the accolades the team received for the work that they've done. I'd like to thank you all for your attention today. Again, it's been a privilege to share this story with all of you and how we're all going to work together to combat antimicrobial resistance going forward.
Thanks, Matt. So we can open for questions, and we already have one here.
Thank you very much. My name is
Make your way to the microphones.
Jennifer Martin. I'm a physician and in my country a clinical pharmacologist, which is also a physician. We spend a lot of time with these drugs. I mean, it's not just a problem for us clinically, but it's a problem for us from a regulatory and a cost perspective as well. Yeah. So I guess one of the issues we've got is just looking at your data. I don't know if you wanted to flip back to the slide where you're looking at the non-inferiority, comparing it with colistin, because colistin is a widely used drug. It's very cheap, off-patent. It definitely does have toxicity, but when you know how to manage it and change the dose or measure exposure, you can actually get the dose pretty good to minimise toxicity. And we use that quite a bit for Pseudomonas in our CF patients. The question I've just got is, when you talk about the non-inferiority, I wasn't actually sure that your trial did show non-inferiority, and maybe we were just missing some data there. But can we have a look at the slide again? Because I kind of thought that was an important problem, only because a lot of issues like these drugs have failed when people have muddled up non-inferiority with equivalence or superiority. And I think you can't really take a non-inferiority trial and then say that it was close to superiority. I think we weren't even sure that we had actually met the non-inferiority margin there. So I just wanted to, if we could go back and have a look at this, because I think it might be worth having this discussion now rather than down the track when it is coming up to the regulatory authorities. So basically across the confidence, so your primary endpoint, I see, so we don't really have the non-inferiority margin there, do we? Because that will help us know whether it was really non-inferior or whether it was actually inferior for the primary endpoint.
Yeah, so for this talk today, I mean, obviously this is a very high-level discussion. I mean, we could do a 2-hour talk just on the analysis of the results and that non-inferiority margin. Happy to chat about that,
okay.
but rest assured that yeah, we very clearly surpassed the non-inferiority margin of what was set with 20%. We actually made the 10%.
Okay, I just felt that there was a lot of sort of positive spin considering the data didn't really show really all that much, to be honest, although it is great that we've got a new drug. But going back to my equity point, when we look at, I mean, Australia's a pretty wealthy country, but if we're looking at the majority of the world where we've got these problems, I'm not sure how they're going to be able to afford these really, really expensive drugs because they won't be reimbursed by a public funder. And I wondered if actually the emphasis should be on a superiority trial to colistin, because otherwise I think people are just going to say, well, we've got colistin, this drug might be non-inferior, but actually it costs a lot more. We're not really sure about the liver toxicity. That could be a problem for us, and it's a huge expense that our public funder can't afford. So that they're just really more questions, and whether when you're thinking about your commercialisation plan, you had considered the sort of cost and the equity issues around that.
Yeah, absolutely. Cost is a big factor in pricing. I'm not going to talk about pricing today, but it's an enormous factor, especially in the space. It's exactly the points that you're saying. There are a lot of antibiotics out there that are cheap. Colistin's one of them. I know if I had a choice, do you want to go on colistin or XACDURO, I would absolutely choose XACDURO.
Yeah, but you're the company guy.
It's really, XACDURO, I mean, colistin's a tough drug. I mean, I think everyone's familiar with colistin and the issues associated with that. But again, our point, we chose colistin as a comparator because it is the most prescribed.
Okay, thank you.
Okay, we have time for 2 more questions, so can we come to the front and then back to the back?
Yeah, thank you. My name is David Kredian from Hungary. My question would be, I've recently read the trial for cefiderocol, which is also against these gram-negative superbugs, for example, for Acinetobacter, as a potential competitor for colistin. And what I found really interesting in that trial is that they found differences for their endpoints if they checked for Acinetobacter bone infection as a pneumonia, originating from a pneumonia, or originating from UTI, or from originating from different points. So perhaps did you check something like that, if there is a difference for your endpoints, if it's like you mentioned just broadly Acinetobacter infections, but is there a difference between a pneumonia or a UTI, and is there pharmacokinetic differences, for example, that could account for that?
Yeah, so that's a fantastic question. So our label is specific to pneumonia because the predominance of patients in our clinical study were pneumonia patients. We also have an expanded access program that we've been conducting since 2020, where we have treated patients that are not pneumonia-based. We have burn victims, skin associations, and things along those lines. Some of that data is published. I believe the burn patient specifically is going to be published shortly. But this is something that's going to be continual post-approval evaluation, and really looking at other sites of infection and how the ability for XACDURO to treat those. So stay tuned. There'll be other data hopefully coming out in the next few years.
Thanks. So one last quick question.
Thanks. I did a 9-step synthesis as an undergraduate, and it put me off medicinal chemistry for life, so a lot of sympathy for your manufacturing guys. So yeah, quick question is, as you alluded to, colistin doesn't get into the lungs, and it's probably difficult to optimally dose it, and you need loading dose, everything. So you kind of had an, inverted commas, easy comparator. So how do we develop the next drug for Acinetobacter when the standard of care is sulbactam, durlobactam?
So that's a great question. And one of the data sets we're also very much looking forward to seeing, I'm going to get to your question in a second, but I'm going to bring this back a little bit to XACDURO. Again, we're developing, or looking to develop a paradigm where you're dosing the right drug at the right time and the right dose. It's not a broad dosing like you would see with the broad spectrum antibiotics, where resistance rates are going to increase rapidly because of that. So what we're hopeful for is, by developing this type of precision targeted therapy, we're hoping that what the data ultimately shows is the resistance rates are much slower than what they might be with a typical broad spectrum. To answer your question, what's the next generation beyond XACDURO? That's going to be hard to, I think, answer at this point. Are you thinking more within the same chemical series, or are you thinking more just in general as an antibiotic?
Well, it's just trial design, because it's going to be so much harder. Yeah, so it's just trial design, because it's going to be so much harder. You're going to have to meet a non-inferiority margin of a drug that's much better than colistin. So what's the next trial? Can we avoid doing any trials, comparative trials, and do things on PK/PD and safety?
I see what you're saying. That's a great question. I think that's definitely worth continuing to think about. How do you do this in a more efficient way? How do you do this continually? Because again, conducting a study like this is complicated, and it takes a lot of courage in order to go into something like that. But to your point, the lessons learned from what we were able to accomplish, how can we apply that to create greater efficiency in trial design going forward for pathogen-targeted therapies? I think is that the question you're asking? Yeah, that's going to take a bit more evaluation, lessons learned from what we did, and there's plenty of lessons learned. But I think to your point, yeah, how do we make it even more streamlined and more specific to other pathogens as well, with different chemical entities?
Okay, thanks, Matt. And just it seems to me that there's a great deal of interest, and it does look that there's a potential new paradigm here for precision therapies for different microbes, and perhaps there'll be a different economic model that appears from that. I remember somebody asking once, why is the life of a child dying with leukemia worth more than that of a child dying of a specific bacterial infection? It's a very difficult question to answer, and yet the economic models around medicines for those two conditions somehow seem very different. And I wish we had a lot more time to debate some of these issues, but sadly we don't. Just before we move to our last talk, can I just ask everybody here who I guess has some interest in antimicrobials, do try and come along to our lunchtime session at 12:30, where we will look at old paradigms in antimicrobial therapy and meet the daughter of penicillin's first patient and learn about the development of penicillin, which was really something that was a remarkable story in its own right and started this whole process. Well, our final presentation today, is from Rebecca McHugh, who is an early career researcher at the University of Glasgow. And Rebecca's research is really interesting because she's looking at making molecules which prevent virulent bacteria being virulent without necessarily killing the bacteria themselves. And I think this offers, again, a new paradigm in antimicrobial therapies where we perhaps have inbuilt mechanisms that will diminish the ability for resistance to come around. So, Rebecca, thank you very much, and over to you.
Thank you. Hi everyone. First and foremost, thank you so much, Mike, and to the organisers for giving me a chance to come along and speak to you all today. I very much appreciate being some of the local representation. So yeah, I work here at the University of Glasgow. I would strongly encourage anybody who's visiting Glasgow to go along and pay a visit to the campus, because it is a very beautiful place to work. So Mike asked me to come along today and kind of give a bit of an overview about recent advances in antimicrobial therapeutics. And as a natural products biologist, really, a lot of my talk today is going to focus on that from a kind of natural products, bacterial natural products point of view. And we have heard so many wonderful talks today about the kind of the further clinical development and policy, but I'm going to focus mainly on before we get to that point in the drug discovery pipeline, and I'm going to speak about where we can actually find new molecules that have therapeutic potential. And so firstly, I'm going to speak about our natural product discovery pipeline. I'll then go on to speak about the problems that we have with this pipeline and how we can improve on those problems by integrating new technologies and also collaboration with other disciplines. I'll then speak about some examples of some new and interesting molecules that are coming out of this development pipeline, and then I'll speak to you a little bit towards the end of the talk about my own work towards antivirulence therapies, as Mike mentioned. So I was wondering if we'd see a similar slide today at some point, but just to go over this, this is the kind of timeline of antibiotic drug discovery, and in the green in the centre you can see this golden age. And really, since penicillin was discovered and Fleming found this petri dish with the mould growing and the mould producing this inhibitory substance that inhibited his culture, we've kind of used the same methods to discover novel antimicrobials. And during this golden age, when streptomycin was discovered, the first streptomyces bacteria were characterised. And streptomyces are really interesting bacteria because they produce a huge amount of medicinal molecules. And when I say streptomyces, there's also closely related bacteria to streptomyces that are also very good at this, as the greater group of actinobacteria. So these multicellular soil-dwelling actinobacteria, and they have this unique and complex life cycle. They have spores. The spores will germinate, then will form aerial hyphae. We'll see that on an agar plate. They look kind of fluffy like fungi. And it's at this point, and then when they go to form spores, that they'll start to produce this whole host of interesting secondary metabolites. And these can be antibacterial, antifungal, even anti-cancer, and and immunosuppressants can be produced by streptomyces bacteria. And here's just some examples of the bacteria and what they can produce. So chloramphenicol, you might get in your eye drops. They come from streptomyces venezuelae originally. Ivermectin, used to treat helminth infections for river blindness, Nobel Prize winning drug, or ivermectin derived from this, is produced by Streptomyces. Orodox, which I'll talk about later on today, which is my antivirulence product, produced by Streptomyces goldeniensis, and another beta-lactamase inhibitor, Streptomyces clavilegerus, produces clavulanic acid, used in augmentin. So really, really talented bacteria in terms of what they can produce. So when we try to isolate new compounds from Streptomyces, we've followed this pipeline for a really long time, where you go out into the field, you isolate soil, because we know Streptomyces are so abundant in the soil, and then we'll isolate strains, we'll grow them in monoculture, and then we'll grow them on diverse media to encourage them to produce different molecules. And we'll then take plugs of that media, if it's on agar, or extracts, solvent extracts of the media, and test them in bioassays against our pathogen of interest. Which sounds all well and great, but all well and good, but obviously we're no longer in this golden age of antibiotic discovery, so there are problems with this pipeline. Because it might take a very long time to get to this point where you can extract your compound in enough abundance so that you can get a nice peak on your LCMS and then do some NMR and identify a structure. And then you might get to that point and find that you've just rediscovered streptomycin, or you've rediscovered penicillin, or you've rediscovered something that we already use in the clinic, and it's a lot of work until we get to this point. Or you could have even rediscovered another molecule that could never actually be used in the clinic at all because it had poor pharmacological properties. And that's something else. You can spend 5 to 10 years on some of these molecules that are really interesting, that come from our bacteria, and you finally get a mouse model up and running, and there's poor efficacy. So obviously there are problems here that the natural products community has tried to address. So we want to address rediscovery and try and limit that. There's a number of ways that we're doing this, by exploiting new ecological niches, and by trying to extract compounds from previously unculturable bacteria. We want to try to repurpose cytotoxic compounds for other things. I'll talk about that a bit later. And collaborate with synthetic chemists and pharmacologists like yourself to try and improve some of the properties of these drugs. So in terms of antimicrobial discovery, we know that many bacteria in the soil are very talented at producing antibiotics, but sometimes it's difficult to actually culture these bacteria in the lab. And so this group here made some new technology known as the iChip, where essentially you take your culture and bury it in the soil that it was originally isolated from. And this means that it's got all the correct nutrients for your strain to grow. And through this method, they were able to isolate a new compound, Texabactin, which seems to be extremely promising against things like MRSA and their tests in the lab. So this is, and it's got a two-pronged mechanism of action, so hopefully good longevity in the clinic. And it's currently in preparation for clinical trials as far as I'm aware. And this is just a good example of how we can deploy new technology in order to try and really search previously unexploited niches for, to find novel chemistry and novel compounds. In terms of antimicrobial discovery, when we look at genomics, it's really helped our discovery of novel compounds in many ways. In 2002, the whole genome of Streptomyces silicolor was published for the first time, and until this point, we were really unaware of just how good Streptomyces were at producing these antibiotics. So we knew that Streptomyces silicolor, the Streptomyces model organism, could produce 4 different antibiotics. And when the genome was sequenced, more than 20 genomic regions were found that were associated with antibiotic production. So these were then coined cryptic or silent biosynthetic gene clusters, and there's now a lot of work going on to try and get these gene clusters activated in the lab so that we can study the natural products and again hopefully take them forward in the clinics. It's also important to note that now we don't actually need to culture the bacteria from the soil to study their natural products. Metagenomics has come so far that we can actually just sequence the DNA in the soil, look for genes within that soil that encode predicted antibiotic clusters and clone those out and try and express them in other species. So some of these developments are really interesting and could potentially really advance our understanding of these products. One example of this is the formicomycins. Now, this work was done down in the John Innes Centre, a huge centre of antibiotic research, where they looked at fungus farming ants, these Tetraponera ants. So they found that these ants, they basically grow, they have a fungal garden and they farm the fungus for their food. And there was actually invasive fungus that was coming in and destroying their nests and using it as a food source. So they found that these bacteria, these ants actually carried Streptomyces bacteria on their chest, and they would inoculate their fungal garden with these bacteria so that they could produce specific antifungals that would kill the invading fungus but leave the fungus that they farmed alone. And so they sequenced some of the genomes of these Streptomyces organisms, and they found that there was many natural product gene clusters, and they found this method, the formicomycins, which again, they've been studying this molecule for up to 8 years, and they haven't managed to evolve resistance in the lab, which means they haven't managed to get a mechanism of action yet. But it's a very potent antibiotic, and it's active against VRE and MRSA. So again, just an example of how you can exploit these ecological niches and these novel environments to look for new bacteria and hopefully new molecules. This is just another example. This is one that my supervisor, Andy, loves to tell. He's like, you know, they can isolate antibiotics from crocodile blood. So, yeah, the crocodiles, they get bitten all the time and they're always in fights, but they don't experience a large number of infections. And so scientists looked at the blood of these crocodiles and were able to isolate novel antimicrobial peptides, and these are being studied now. So again, exploiting these ecological niches to try and look for new chemistry and new molecules that can help us in this search. So just to speak about my work a little bit, I work on drugs for Shiga-toxin-producing E. coli. Now, resistance isn't necessarily the main issue with this. Shiga-toxin-producing E. coli, they encode these phage Shiga-toxins that are extremely cytotoxic, and they can cause severe damage to the kidneys and the brain in a syndrome that's known as hemolytic uremic syndrome, or HUS. And the issue is, if you give traditional antibiotics, because that toxin is encoded on a phage, that phage will be activated, and traditional antibiotics actually cause the bacteria to produce more toxin and make it more likely that the infection might stay in the gut, but the toxin will damage other organs in the body. And so we have this common serotype in the UK, which is E. coli 0157H7. And actually in Scotland, we have a particular problem with these kinds of E. coli with around 3 times higher rates than the rest of the UK. And as I say, it's a particular issue because there are no suitable antibiotics for the treatment of this infection. And so we aim to use antivirulence therapies to treat these infections. Now, this is not like traditional antibiotics because we're not actually aiming to kill or inhibit the growth of the infecting bacteria. We simply want to prevent those bacteria from being able to colonise or harm the host. And in the case of pathogens of the gut, it would allow the immune system to kick in and remove the pathogen. And obviously the gut has a natural cycle of removing waste as well that would help get rid of that pathogen. In theory, it would have positive implications for the development of AMR. If you think about why bacteria become resistant to antibiotics, it's because if they don't, they won't survive. So there's a really strong selective pressure. But this isn't the case with antivirulence compounds. That same selective pressure for resistance isn't there. So we hope that these compounds could be used where traditional antibiotics aren't able to be used. So in my case, we aim to target the type 3 secretion system, and this is an injectosome that injects proteinaceous effectors into the host cell and allows the host cell allows the bacteria to initially colonise the host cell. So in theory, by blocking this process, the bacteria will just be removed from the gut. And the molecule I work on specifically is Orodox. It's a molecule produced by Streptomyces goldeniensis. And this is a case of repurposing a molecule because it was discovered all the way back in 1972, and it was originally investigated for its ability to inhibit the growth of gram-positive organisms. But it didn't make it to the clinic. It was, like a lot of shelved antibiotics at the time, used as a growth promoter in poultry. And actually we understand its mechanism of action in this context very well. We know that it binds to elongation factor 2, which is a protein essential in the protein synthesis cycle. So by blocking this, it causes cell death in gram-positive organisms. And actually in 2011, it was pulled out from one of these huge compound screens as a potential inhibitor of the EPEC and EHEC type 3 secretion system. So this is where my work came in. We decided to look at whether or not the compound could block infection. So you can see in this bottom panel here, we've got our infected cells on the left-hand side that are untreated. And these GFP tagged, the EHEC bacteria, are using their type 3 secretion system to form these tight colonies, microcolonies on the epithelial cells, and they're forming actin pedestals or lesions, which are really characteristic of EHEC infection. When we add Orodox, although the bacteria are still there, they're still present, they aren't able to form these colonies or form these actin pedestals. So we knew that this compound was acting, and we knew this compound was fairly potent in this activity. And when we quantify this, we see a 3-fold reduction in the number of cells infected and a greater than 7,000-fold reduction in colonization efficiency. So yeah, we're quite confident in the ability of the compound to do this job. And it was also really important to us to make sure that Orodox didn't induce Shiga toxin production. So if you look at ciprofloxacin, for example, this is a gene reporter assays where we're looking at the expression of certain genes. And you can see REC-A, which is a stress response gene, and this activates Shiga toxin eventually. Ciprofloxacin drives the expression of this gene through the roof, whereas we don't see activation with Orodox. And when we Western blot for this, we see the presence of Shiga toxin in our ciprofloxacin treated sample, but not in Orodox that are untreated. So we're very hopeful that this could be taken further down this drug discovery pipeline. We then developed an animal model to test whether Orodox could protect mice against hemolytic uremic syndrome, this Shiga toxin mediated disease that's associated with STEC. So E. coli 0157 and similar bacteria, they don't readily infect mice. So we use a murine model, Citrobacter rodentium, that carries the same type 3 secretion system as a surrogate model. And we actually have one of these strains with Shiga toxin to mimic what would happen in a STEC patient. And so you can see that the mice, after a few days, they start to lose weight, and they have to be culled at that point at 15% weight loss. And you can see really high colonization. And when we look at the histology, you can see clear signs in the histology of HUS that include epithelial migration into the tubules of the kidney. And so we had quite a nice model here, and we wanted to test whether Orodox could improve the condition of the mice in this model. And so we dosed our mice with a Citrobacter producing Shiga toxin, and we started to treat with Orodox 8 hours later, and they got 1 dose per day. And over the first 7 days, all the animals, even in the PBS control group, lost weight. That's just probably the stress of the oral gavage on the mice. But our Orodox animals regained the weight by the end of the experiment, whereas our PBS animals gained the weight, a little bit of weight back, and then continued to lose the weight. And actually, our survival in our Orodox treated animals was 100% compared to 50% in our PBS treated animals. So this was a really positive result for us after working on the compound for such a long time. So just in summary, and probably over to summarise the whole morning really, AMR is one of the key challenges that we face today and in the future, and investment in education are essential. We want to use novel technologies and multidisciplinary collaborations in order to accelerate drug discovery and the advancement of these drugs down the discovery pipeline. We want to use this novel technology and incorporate it into this pipeline. And new strategies such as antivirulence may be able to supplement antibiotic therapy as we move forward into the future. And just quickly at the end, in terms of education, this is Dr. Dirt. This is a game that I developed with my colleagues at the University of Strathclyde and the Game Doctor. And this is a game you can play. The website's there. You can download it on an iPhone or an Android. And it's about how we actually discover drugs. So you can go take your test tube into the sea or take your test tube to different environments and isolate the bacteria and run them on the mass spec and see if you find anything interesting there. So suitable for ages 8+, just if any of you do engagement activities in this kind of context. And just finally, I'd like to thank everyone at the University of Glasgow, my colleagues at Strathclyde as well, my funders, the Medical Research Council and the Microbiology Society. Thank you for listening, thank you for inviting me, and I'm happy to take any questions.
Thank you, Rebecca. So we have time for some questions. Usual, come to the microphone if you wish to ask one. I just ask about Orodox. Is its safety profile good enough, do you think, to contemplate for clinical use?
Yes. So we've heard this via word of mouth rather than any official documents, that back in the day it got through phase 1 of the clinical trials, but it was an efficacy issue, so it didn't get past phase 2. But so it wasn't particularly good against the other compounds available at the time at killing the gram-positives in vivo, especially in humans. In our mice, we see a good effect. We'll try and optimise that study a little bit more over time. And we've done all the kind of in vitro studies, so I'm hoping that it's okay at the moment. In terms of, there are some issues with it. Solubility is not great. We have to give the mice the compound in oil at the moment, so we've got some people over in chemistry on board that are going to help us try and tweak the molecule a little bit to help that side of things.
Just very quickly before we go to the question, so word of mouth said that it's got through a phase
Yeah.
1, which is great, but I just wonder, is this a compound which would be in Laura's database? Because to me it seems that if there is phase 1 data that says something's safe, it's a great pity that we don't know that formally.
Yeah.
And it's how to get that information.
So Orodox itself isn't, but there's other alpha-mycin compounds, which are like chiromycin, that are in the database. And I've tested them all in this model, and Orodox is the best. It's basically just got one extra methyl group. And chiromycin, actually, just one methyl group's the difference. Chiromycin actually kills the E. coli, which we don't really want. So yeah, it would be really, really interesting. And the compounds themselves are safe, as far as we know. But my specific variation on that, we might need to do some more work on.
Great, thank you. So there's a question.
Yeah, so my question was sort of related to the answer you just gave about the Orodox. How did you decide that it should be given once a day, and how did you decide on the dose, and what's the future optimization that you might be doing on the PK/PD side?
Yeah, so we decided on the dose based on the dose that was given in one of the original papers, and it seems to work. There was 2 suggested doses in the previous paper. It was 2.5 mg per kg or 25 mg per kg, and we used the lower dose and it seems to work. The reason we used the lower dose is because it's quite expensive to buy at the moment, but the other side of my project is working on the optimization of Orodox production, and it's producing. I didn't speak about it today, and we're getting there with that. So I'm hoping by the end of this ProStock project we'll be able to do a lot more optimization, because we'll have a lot more compound available, if that makes sense. Thanks.
And there is another question here, or a comment.
Hi.
So I don't think you can even hear me with this microphone.
I can hear, there's a speaker here.
I'll shout. Just to say about Antibiotic DB, I'm really glad you mentioned it, because it is incomplete. That's one of the reasons we have a poster here and at other conferences. So we encourage people, if they find anything missing that should be there, to fill in the form on Antibiotic DB, just to say that it's all going to be relaunched later this year, early next year, to be much easier to use. but there are just over 2,000 entries at the moment, and one of the areas that we are substantially lacking in is natural products. So if there's anyone here with information, please do add it to Antibiotic DB because it helps the community.
That's something we can definitely do, and working in the rest, I work with natural products people at Strathclyde, so I can recruit them to help out as well.
Fabulous, thank you.
No problem.
Well, that's great. And almost to the minute, we have finished on time. So I would just like to thank all of the speakers for keeping to time, but also for injecting some optimism that we are not doing so badly against the problem. We recognize it, and innovations are appearing, I think both at the scientific but also at the societal level. So let's hope that we're going to be able to keep the beast of antimicrobial resistance at bay. So I'd just like to thank everybody once again. Please do come back at 12:30 for that special session at lunchtime. Have a quick refreshment prior to then. And I would just like to thank everybody for a wonderful morning here. Thank you.